Okay, so here's a diagram of a type 4 virus, and a couple things here. This ends up being a single-stranded RNA that's inside of it. This is also the type of virus that has an envelope around it, so we've got our capsid in here. The protein that's surrounding the red is our DNA, okay, or excuse me, our single-stranded RNA, and then it's surrounded by a membrane. And so what we're going to do here is we're going to display what happens when we have a type 4 with a single-stranded RNA that's used as a template to make messenger RNA. We are also going to talk about how an envelope is made, okay, to go around this bacteria. So first of all, no details on this, but we're getting our virus inside of the host. Okay, so an animal cell here. Once it gets inside of the host, it breaks apart, and we have our viral single-stranded RNA is released. Okay. Now, something that they're not showing here is that inside of this virus, as well as having the single-stranded RNA, it's going to have some sort of an enzyme that its job is to take the RNA, the viral genome, the RNA, and basically use it to make messenger RNA copies of itself. Okay, so that it can, anyway, but that enzyme is going to be inside of here. We're going to see this when we look at the HIV virus on the next slide as well. Okay, so anyway, we have this viral genome, the single-stranded RNA. That's going to be used to make a template. Okay, it's going to be used to make, or as a template to make messenger RNA. Okay, so this, again, we're kind of using it as if it's DNA, but it is RNA, and so it has to be a new virus, because inside of a cell, animal cells don't normally have any kind of an enzyme that would take RNA and make a messenger RNA copy from it. Okay, so there has to be a viral enzyme that does that. Anyway, so we have this messenger RNA that can do what it does. We'll get back to that in a second. The other thing that happens is this RNA copy is going to be used as a template to make a complementary copy, which would be identical to the original genome. Okay, so if we kind of go through complements here, the messenger RNA ends up being complementary to the genome, which means we can use it as a template to make more genome, okay, for the virus. And so that's a step that's happening over here. We're going to make a bunch of copies of the viral genome. If I step back to the messenger RNA, there's going to be genetic code here to make all the capsid proteins so that we can surround our genome RNA with the protein, with the capsid. But then there's also going to be genetic sequence here to make glycoproteins. And so what's going to happen is this messenger RNA is going to attach to ribosomes that are on the rough ER, okay, and it's going to make glycoproteins in the rough ER. If we go back a bunch of chapters, this should sound familiar. Those glycoproteins, we're going to put them in a transport vesicle, which is going to transport them to the outer membrane. This vesicle will actually become part of the outer membrane, and then the glycoproteins that are on the inside of this will then end up on the outside of the cell membrane. So we have a bunch of glycoproteins on the outside of the cell membrane. When we have our assembled viruses with their viral genome in the middle and then the capsids on the outside, this is basically going to be exported here to the cell membrane, the host membrane, and it's going to get bound by the host membrane that has all these glycoproteins on it. Okay, so that's how we get our envelope. The purpose of those glycoproteins is they are the way that this virus, when it goes to a new cell, that's the way that this virus gets accepted by the new cell. They give a signal to the new cell that basically says, accept me, okay, or take me in, and that's how this will attack the new one. But so that's the idea of an envelope being made. It is also showing this type 5 classification of viruses, where our RNA is going to be used to make messenger RNA, and then there's going to be another process that makes more copies of the genome. So a couple things there. Next we're going to look at